the recording and pass the mic off to Dr. Puxeddu.
Hello, hi everybody. Thank you for joining me and thank you for the introduction, Haley. So yes, today I'm going to talk about my latest work, which has the title Leveraging Multivariate Information for Community Detection in Functional Brain Networks. And before I really start, this is the paper. It was a collaborative effort with my previous co-workers at IU when I was a postdoc. Yeah, it was a teamwork with Maria Pope, that is now in University of Chicago, Thomas Bartley, who is now in University of Vermont, and Joshua Faskowitz at NIH, and my ex-advisor, Ola Sporns, who still is at IU. Okay, let's get into brain networks. So, as you might already know, the brain can be modeled as a network. So we have different brain areas that are our nodes, and they can be connected or not to each other, and that forms our brain network. Okay, and that is the case of we can have anatomical connections, and that forms our anatomical brain networks, or we can have functional connections. That is, for example, two nodes display brain activity that is highly correlated, and that is a functional connection. Okay, so the interesting thing about the brain networks is that they exhibit a complex organization. So their topological organization is not random, is not regular. We don't have lattices as brain networks, thankfully. It's something in between. Okay, it's a complex system. And one feature of this complex organization is the presence of modules or clusters. And these are groups of nodes that are highly interconnected to each other. So for example, in the case of functional brain networks, the brain activity that we register on top of these nodes is highly related. And so we have high similarity in brain activity in nodes of the same community, and then lower similarity of brain activity across communities. And these features of the brain networks, it's quite nice because it allows for a balance between segregation and integration of information, which is what allows for emergent properties like cognition and complex behaviors. And so one goal of network neuroscience is to describe and identify this modular structure in the brain and see how it can be associated with cognitive performances, development, aging, or brain disorders. So how do we find these modules in the brain? At the state of the art, so in this talk, in general, there are many definitions of communities in a network. In this talk, I'm going to focus on assortative communities, which is what I've been describing so far. So those communities where nodes are highly interconnected to each other and show lower connectivity outside of them. So how can we identify those modules? At the state of the art, there are two main methods, two main classes of methods, I would say. One is, on one side, we have diffusion-based algorithms like InfoMap, where we have a random walker. Those are based on random walks. So basically, we identify as communities those groups of nodes where a random walker gets trapped. And on the other side, we have density-based algorithms like modularity maximization, where we identify modules as those groups of nodes where we count a lot more of connections than outside of the modules. What both classes have in common is that they both use pairwise interactions to identify this modular structure. And in doing so, they disregard a whole world of higher order interactions that involve 3 or more nodes at the same times and are as equally as important to fully characterize the brain dynamics. So the question that we wanted to answer with this work is, can we, first of all, can we provide a description of the modular structure that accounts for higher order interactions? And consequently, can we implement a community detection algorithm that find a modular structure that accounts for these higher order interactions? So this is our general aim. So we want this new definition of modular structure that account for higher order interactions. And we suggest this new definition using notions of information theory. So again, here we have our brain, we have nodes, which are brain regions, and we have time series associated to each one of these brain regions, which is their brain activity. We can compute from this time series 2 types of information. So on one side, we have redundant information, which is the information that is copied across the time series, sorry. And then we have synergistic information, which is that information that is accessible only when I look at the whole set together, at the whole set of time series together. And so ideally, what we want to find is a community structure that displays high redundancy within modules. So the time series depicting the brain activity of nodes within one cluster show high redundancy. So they carry the same information to the system. And then on the other hand, we want to have high synergy between modules. So when we integrate the information from nodes belonging to different communities, then we have access to a new type of information that we cannot reconduce to either of the modules from when we're picking the brain activity. So this is the new definition of modular structure that we suggest. So then the question is, how do we find this kind of community structure in the brain? Well, the idea behind it is that we want to maximize redundancy within modules for the reasons that I explained just now. And so to do that, we use total correlation as a proxy of redundancy. Total correlation is a measure derived from entropy and is low when the variables are independent and is high when instead the joint state of the whole system has low entropy. So high values of total correlation indicate high redundancy of the time series that the subset of time series that I'm considering. So what I can do here is, so to find these communities in the brain, I start by building this leaf diagram here, which for each subset site, so for each dimension of time series that I'm considering, returns the average level of integration that I expect from a subset of that size and the maximum level of integration or total correlation or redundancy that I can expect from that subset size. So let's say, for example, I take all the subsets of 50, so that are comprised of 50 time series. I can compute the average level of total correlation and the maximum level of total correlation. And then what I can do, what the algorithm does is it starts assigning nodes randomly at the beginning to modules. And for each modules, it computes the total correlation. And we can see where these total correlation values lay on this leaf diagram. So because we partition the network randomly by assigning nodes to clusters in a random way, we expect the total correlation of these modules to be close to the average level of integrations. Okay, but what we want to do is we want to optimize the redundancy within modules. So we want this value of total correlation within modules to be as high as possible. We want it to deviate as much as possible from the average level of integration for subsets of equal size. And so what the algorithm does is it starts reassigning nodes to modules in order to increase the distance between the total correlation within module and the average level of integration for subsets of equal size. So here you can see the random dots are the total correlation values at the beginning on the random seed partition, and here the red dots are the total correlation values of the modules after the optimization process. And the optimization happens with a simulated annealing algorithm. And so this is a new quality function that we optimize. We optimize the difference between the total correlation within modules and the average level of integration that I expect for subsets of equal size. And this is an example of optimization. So we start from a random partition of the networking of the brain into 5 modules. And this is what we obtain after the optimization. And just by visual inspection, you can see that the modules that we obtain are pretty much contiguous, spatially contiguous. And this is something that we kind of expect from the brain because it has physical constraints. They're also symmetrical with respect to hemispheres, and this is also something that we like because we expect this kind of symmetry across hemispheres. So just by visual inspection, we were pretty happy with the algorithm. However, we wanted to see how it compares with state-of-the-art methods and what insights it brings that we didn't have before. So first of all, we compared our new quality function that we call t-score to the state-of-the-art methods, so modularity basically. So we took our functional brain network, we partitioned it using modularity maximization. And in doing so, as you might know, modularity maximization depends on this parameter gamma that defines the resolution of the partition. So we ran modularity maximization by tuning this parameter in order to obtain coarse and finer partitions. And on top of these partitions, we computed our quality function, t-score, and this is what we obtain. So we obtain a high correlation between t-score and modularity, which means that partitions that are highly modular also tend to have modules that are highly redundant. However, this correlation is inverted. What we saw is that this correlation is inverted if we focus on partitions of equal size. So let's say I focus on partitions of all the partitions that have 10 modules or 7 modules. At that point, the correlation between modularity and t-score is negative and very much close to -1. So it's pretty much it's very strong. They're very strong and anti-correlated. So we have this sort of Simpson paradox where overall, across all scales of resolutions, t-score and Q are highly correlated, but then if we focus on single scales, this correlation is inverted. So that means that there is something that t-score captures that is not captured by Q and vice versa. So for example, this is for 7 modules. These are all the partitions with 7 modules, and you see the partition with higher Q, with highest Q is the partition with lowest t-score and vice versa. And what we did at this point is we took again, the partition with highest Q and the partition with lowest Q. And we saw that what changes is that t-score gives higher points, is higher in partitions that are more symmetrical between the two hemispheres and have lower variance of module size. So we expect that for example, among all the partitions with 7 modules, here on this side of the graph, we have partitions that are more symmetrical and their clusters display less variance in the size. And these are rewarded by t-score. So this is something that is different from t-score and Q. And that we can access by looking at higher order interactions. Then we also asked how our partitions relate to the canonical system, so the Yao systems. So we took our functional connectivity metrics. We partitioned it using our new algorithm, and we observed how similar were the partitions that we obtained to the Yao systems. We observed a peak in this similarity for partitions with 7 modules, which is something that we expect because this is the number of the Yao systems. And but then we also wanted to see locally how they overlap. And what we saw is that there is high overlap at the level of the sensory areas, while there's a redistribution of the nodes belonging to the transmodal cortex. So as you can see, for example, the visual system is pretty well captured by module 1. You can also see here visual system, module 1, they almost perfectly overlap. The sensory system also is captured by module 2 and module 4, although in module 4, there's also part of the ventral attentional network that bleeds in. And this is something that we expect because there are, I mean, we did not completely expect it, but there are parcellations that, for example, put together nodes of the ventral attentional network with the sensory motor system. So this is not totally surprising. So again, this sum motor system is very well captured by modules obtained with the t-score optimization, and same for the dorsal attentional network. But what was different was the partition of the transmodal cortex. So as you can see here, the nodes belonging to the frontoparietal system and the DMN are completely redistributed across 3 modules that are module 5, 6, and 7. So partitioning functional connectivity by taking into account higher order interactions leads to a new organization of the transmodal cortex. And in the future would be very interesting to see how this new organization is related to cognitive performances or how it changes in brain disorders. We also looked at how this score varies across spatial resolution of the partitions. We observed that it decreases. And then, so which means that we observe modules that are that have less redundancy within them as we go to finer resolutions. And we also observed how the synergy across modules varies with spatial resolution. And so to compute that, we used as proxy the O information, which is also derived from entropy, and this is and total correlation, and is a proxy of synergy, and it is negative. When it is negative, it denotes synergy among the time series of the subsets. Here, what we did is we took time series of nodes belonging to different communities, and we computed the O information on this time series to see what's the to have a proxy of synergy across modules. And this is what we display with the purple box plot. In the gray box plot, we have a null measure of it. So we took a random series randomly, disregarding the community structure, and we computed the information from them. So what we observed is that the information increases with the number of clusters, but also the observed model deviates more from the null model as the cluster number increases. So what we wanted to do now is we wanted to see if there is some scale where we have a sort of optimization between the segregation of information and integration of information. And so we define this index, which is just one of the many indices that can be proposed to find that this balance between segregation of information that is that we can see through the total correlation within module, and the integration of information that we can see through the O information between modules. And this is the index that we defined that we called segregation information balance, and is the product between these factors. And so the idea is that we want all these factors to be as high as possible. We want TC score, so redundancy within modules, to be high, and we want this term to be as high as possible because we want the O information across modules to be low. And then we also want the effect size, so the difference between the observed O information and the null information to be high. So if we compute this index across spatial resolutions, we observe this bell-shaped distribution, and we found that partitions that have between 7 and 9 clusters are the partitions that show the highest balance between segregation and integration of information in the brain functional connectivity. We also wanted to characterize locally these partitions. So usually in network science, when we want to characterize the modular structure locally, we use the participation coefficient that gives you an idea of the diversity of the connections. And so, for example, if we have a node that has low participation coefficient, that means that it's very tied to the cluster it belongs to. Vice versa, if it has high participation coefficient values, it means that it shows a diversity of connections. So it has connections throughout the network. Here we want to characterize nodes in a similar fashion to see what are the nodes that have higher and lower topological specialization. However, unfortunately, information theory measures cannot be computed locally. So we cannot have local measures of redundancy or information at the node level. So what we did is we introduced another index that we called relative integration coefficient that is defined as the ratio between the difference between the dollar correlation of module M minus the dollar correlation of module M if we take out node I. And then here we have the dollar correlation of the whole network minus the dollar correlation of the whole network if we remove node I. And so we have high values of relative integration coefficients, and specifically values that are greater than 1, when we have high topological specialization. So when the node I contributes a lot to the redundancy of its own module, because it means that if we take it out, we have this term that disappears, and this should be higher than 1. This ratio should be higher than 1. Vice versa, if we have lower topological specialization, relative integration coefficient values that are lower than 1, it means that there is low topological specialization because this ratio tends to be lower than 1. It means that, yeah, these two terms tend to be pretty much similar, and so the ratio decreases. So we computed this relative integration coefficient for each node, and we found that there are a few nodes that have high topological specialization, so they contribute a lot to the redundancy of their own modules, and those are located mostly on the sensory system. So these are the nodes in green. So there's a visual system, some other motor system. And then there's a tail, a left tail of nodes with low relative integration coefficient that show low topological specialization. And these are mainly located on the transmodal cortex. So there are nodes of the attentional networks and the DMN and frontoparietal networks. Okay, last part of the work, we observed how our redundancy-dominated modular structure changes across the lifespan. So we use the NKI dataset. We took functional connectivity networks from the NKI dataset, which comprises subjects between 6 and 85 years old. And for each one of these, we ran our algorithm to find redundancy-dominated modules, so redundancy-dominated modular structure. We observed how our quality function changes with age, and we saw that it decreases with age. So when we get older, the modular structure that we find is displays lower t-score. So the modules have lower redundancy. And similarly, what we observed is that the O information across modules increases with age. So these two results point to the fact that brain modules become less segregated with age, right? So the redundancy within modules decreases and synergy across modules also decreases. So we have this less defined modular structure associated with age. We also computed the relative integration coefficient. And if we look at the average across all nodes, there is a decrease. So that also points to the same directions, to the fact that as we age, there's lower topological specialization. So there's a less segregated modular structure of the brain. But what was interesting here is that we unpacked this relative integration coefficient for the canonical systems, and we saw that what drives this loss of segregation is the fact that the sum of the motor system and visual system become less redundant with age. So the regions, the sensory areas regions that display the highest level of topological specialization are those that drive the loss of segregation with age. And lastly, we also wanted to see how these redundancy-dominated modules change across years. We wanted to see what are the regions that tend to reconfigure the most across age. And so what we did is we split the data set into age bins, specifically made of 10 years. So we have the first age bin that comprises people from 6 to 15 years old. Then the second bin comprises people from 16 years old to 25 years old, and so on until we have the last bin that comprises people between 76 and 85 years old. And we computed the nodal flexibility, which is how many times nodes change their module assignment across bins. And we found that the regions from the sensory areas, so some of the motor system and visual system, tend to be pretty stable throughout the lifespan. So their node assignment does not change. The regions that change more community assignment are those located in the transmodal cortex. So, and specifically, and the attentional networks. So yes, this was the last result. So in conclusion, in this work, we provided a new definition of moral structure that accounts for higher order interactions. We also implemented an algorithm able to find this new modular structure. And again, it's a modular structure where we observe high redundancy within modules and possibly high synergy between modules, because in this way we can maximize this balance between segregation and integration of information. We characterize the role of the nodes in the network with respect to the new modular structure, and we did that by suggesting this new higher order version of participation coefficient. And lastly, we described how these redundancy-dominated modules change with age. And so this is the end of the talk. Thank you for your attention. And again, thank you to my collaborators, Maria Thomas, Josh, and Olaf for helping me.
Thank you so much, Maria. So we have time for questions. I'll give people a few seconds to gather their thoughts and either send a question in the chat, let me know if you wanted to read it out loud, or you can unmute yourself and ask Maria yourself if you'd like to. So any questions from the audience?
I may have one. Hi. Hi.
Hi.
Can you hear me well? Yeah?
Yes.
Yes. Okay. I'm Francesca. Thank you for the nice talk. I've been working on these topics only like in the last months, so I'm not an expert at all. I'm trying to learn. I come from mostly like purely methodological background. So my first question is, so you said you're looking for modules by maximizing the redundancy, and you do that by using TC as a proxy of redundancy, right? But at the beginning, you also said that the idea is to maximizing the redundancy within and maximizing the synergy between. So I'm missing a bit the connection. So do you expect from the beginning, like from a how to say a prior like point of view, to maximize redundancy within modules and at the same time to maximize the synergy between the modules? I don't know if the question is clear, but
Yeah. So yes, ideally what I want is a partition where I have high redundancy within modules and high synergy between modules. We defined our algorithm to maximize the redundancy within module. And then we obtained this quality function TC score. We hoped that the partitions that we obtained by maximizing their redundancy within modules also led to high synergy between modules. However, we did not put the synergy between modules into the quality function. So we did not maximize at the same time redundancy within modules and synergy between modules. But yeah, so this is something actually we thought many times about changing the quality function in order to take account from the beginning synergy between modules. But yeah.
I don't know if just a few. Uh, you also showed some results on the O information, right? And that was also related to the redundancy, right? Because it was positive, or am I missing?
Yes, we did. So, okay,
Mm,
we cannot zoom too much into that, but
I can see
only,
quite well, no worries.
yeah. So only, yeah, we did not obtain negative or information values except for when we considered uh, mm, partitions made of three modules. So the thing is that this is not something totally surprising, right? Because there's high redundancy across time series of FM across fMRI time series.
Yeah.
Oh, so the higher is the number of time series where we compute the O information, the higher is the chances that we obtain positive values of O information. So only when we took three time series, we obtain negative values. So yeah, we cannot say that we have synergistic uh, information across modules. Um,
Okay, no, but only because I'm finding something similar in a sense. So with what I'm doing right now, I can find like a redundancy in a sense, but synergy is basically no redundancy for what I'm never like really. So
hmm.
yeah, okay, no, that's good for me actually because then
Are you
maybe
also
it's
working with the fMRI time series?
yes. But the
So
first,
yeah,
yeah, yeah, resting state fMRI
yeah.
for the
So
Human Connectome Project, basically.
okay, that's the same. So yeah, the thing is that those are like there's a lot of correlation among those time series. So it's really hard to find
Mhm.
12 time series that are totally synergistic. But yeah, this is there's work from my collaborators, Marie and Thomas, where they look into synergistic information in this data. And yeah, so I suggest you to follow their work too, because their analysis is more on the synergistic aspect, and there are methods with which you can look at that synergistic information even in the data set that we're using.
Okay, thanks. Thank you.
Mhm.
Right. Any other questions from the chat? I have a couple of questions, but I also know Maria, and so I want to make sure that people to whom Maria's work is a bit newer have a chance to ask some questions. Give people a second. While people collect their thoughts, maybe I'll ask one question. So it's interesting that you get like 7 to 9 modules is when you see the highest SI balance, sort of in line with the canonical Yao systems. I have sort of like a two-part question in response to that. Um, how many nodes were in the parcellation that you were using, and did you look at different sizes of parcellations? And to what extent do you think this like convergence on like 7 to 9 modules is like related to the math of calculating the different communities and optimizing the quality functions for different communities? To what extent is like the 7 to 9 related to the number of nodes that you have in your parcellation?
Yes. So, to first answer your question, we have 200 nodes in this parcellation. We did not test the algorithm on different parcellations. We tested on another data set, the MIGA data set, but using the same parcellation. So we found similar results, but yeah, the number of nodes was the same. I do expect that there is some mathematical relationship between these numbers and the number of nodes. Um, yes, so maybe I have a slide that is hidden. Wait. So here we have, well, no, never mind. This is not related to that. So, but yeah, I do expect these factors maybe to be related to each other, but I'm not sure because I haven't tried. So yeah.
Thank you. Yeah, it was just interesting to me that you get like roughly the same number of modules. And so like is it a math thing or is it a brain thing? Maybe both.
Maybe it's a brain thing, but what's new here is that we are suggesting a new way of partitioning these nodes. So again, it would be interesting to see how this new organization of the higher order association cortex relates to development, aging, or cognitive performances.
Yeah, for sure. Any other questions from audience members? There's more that I could ask, but I want to make sure that other people have a chance to voice their questions. Okay, maybe I will ask another one. So I think your aging results are really interesting, and also in line with like some of the other existing literature about like an increase, like more like de-differentiation of different brain systems with age. I thought it was really interesting that you showed that this seems to be driven by like somatosensory systems, like somatomotor systems, and visual systems becoming less redundant with age. And so I kind of want to ask you to speculate as to why you think it might be those systems that are driving um that are driving this result. Do you think there's like something about like the cell types or the genetics or something? I don't know. I'm curious to hear what your thoughts are about why you're finding this result.
Yeah, so that is so I think what's interesting about these results is that, so on one side, we have the sensory areas that drive this loss of segregation with age. At the same time, we have also the transmodal cortex that reconfigures the most with age. So it's like a combination of these two things that it's nice to look at to see what aging means for the brain, right? So but I don't know, maybe I could speculate that because the sensory areas were the ones with with highest topological specialization, maybe the brain as we age goes to a level where like it flattens in a way, like maybe all the regions tend to have the same amount of topological specialization. So, but I like for genetic causes or cellular causes, I don't know. But I think, yeah, again, I think what's interesting about these results is that there's not a like a brain system that whose behavior is associated with age. There are different moving pieces that contribute to the changes of the brain topological organization throughout the age. And so on one side, we observe this loss of segregation in the sensory areas, but then at the same time, we have this reconfiguration of areas that already had lower topological specialization. So yeah, but why it happens, like genetic causes, I don't know. That's something one could investigate. Yeah.
Very cool. Thank you. Um, I'll give people another chance. We have a few minutes until the end of the hour, so I just want to make sure that if anybody has any questions, they can ask, or comments or compliments. The figures were beautiful, Maria. I'll give you a compliment.
Thank you. Thank you.
Yeah. Um, I'll bring us back to a point that we were sort of talking about before the talk officially started. So Maria recently started a faculty position, woohoo, at Virginia Tech, which means you'll be hiring people for your lab.
Yes, both graduate students and postdocs. So if you are interested in working with me, please email me. This is my email. I'll accept grad students from next year, so fall 2026, and postdocs. There's more flexibility for them. So if you're interested, shoot me an email.
It would be a
Thank
great
you for
opportunity.
reminding me that, Haley. I was forgetting.
Yes, of course. Yes, you're great. I want the best people to work with you, so I want to make sure that we get the message out broadly.
Thank you.
Yes, of course. So if there aren't any other questions, maybe we call it. Thank you again so much, Maria, for giving the talk, and sharing your work with us. We'll be posting this recording on YouTube in the next week or so, if you want to come back to it. But I'll go ahead and
Thank